As was briefly discussed in the video, the YouTube video that you watched, and as was mentioned in the PowerPoint on amino acid chemistry, there are various levels of amino acid organization within a protein that contributes to its structure. As we said in class, there is the primary structure, which is the sequence of amino acids within a polypeptide chain. Secondary structures are local structures. Tertiary structures are the three-dimensional structures or three-dimensional shape that a protein takes when it's completely folded, and a quaternary structure is the three-dimensional shape of a protein complex, meaning that there are more than one protein that associate with one another to create the quaternary structure. We're going to focus this discussion on secondary structures, those local structures within polypeptide chains, again, within a single polypeptide chain. And there are two flavors of these secondary structures. The first type of these secondary structures is an alpha helix. I want to look at each of the images on this slide, starting with the one that is on the left. This indicates that an alpha helix is in fact a helical structure, and like DNA helices, alpha helices within polypeptides are right-handed. So we have right-handed alpha helices. An alpha helix, also is indicated on that left image, have 3.6 amino acids per turn of the helix. This image says 3.6 residues. Another word for an amino acid is a residue. So there are 3.6 amino acids per turn of an alpha helix. Looking at the middle image, the red sort of fuzzy lines between atoms within the alpha helix are hydrogen bonds. Hydrogen bonds maintain the structure of an alpha helix, and these hydrogen bonds are between atoms within the polypeptide backbone. I highlight here an oxygen associated with the carboxylic carbon of the first amino acid in this particular alpha helix, and the amino nitrogen group of the fifth amino acid within that alpha helix. Again, alpha helices are maintained by hydrogen bonds between atoms within the polypeptide backbone. That allows for the amino acid side chains to be free from these hydrogen bonds and therefore free from participation within the alpha helix. On the rightmost image, each of these highlighted structures is an amino acid side chain, and you can sort of see that they project out from the yellow alpha helical backbone sort of on the surface of the alpha helix. Because the amino acid side chains are not involved in maintaining the structure of the alpha helix, any amino acid can be part of an alpha helix within a polypeptide, with the exception of proline. Proline is an exception because the side chain itself sort of circles around and becomes bonded to part of the backbone, and that creates, as I've said in class, that creates a kink in the backbone, and that kink makes proline unsuitable for participation in an alpha helix. Looking at an alpha helix from another direction, here we are looking down on to an alpha helix, and each of those red spheres represents an amino acid side chain. This confirms that those side chains are projecting out from the center of that alpha helix. If we know the chemical character of the amino acids that are involved in an alpha helix, we also know the order in which those amino acids are going to occur, again, looking down on an alpha helix. At the very top of this, what is called a helix wheel projection, we see the first amino acid of the alpha helix. Because there are 3.6 amino acids per turn, we know that the second amino acid is a certain distance from the first, the third a certain distance from the second, and so on around the helix. So we can take an amino acid sequence and label each of these dots, I guess, with the amino acid, and that might tell us something about the chemical character of the alpha helix if we know the chemical character of those amino acids. Let me give you an example. Here we have a couple of helix wheel projections with amino acid sequences underneath of them. The amino acid sequences are listed in single letter codes, but on the left-hand side, I have a little legend identifying the chemical character of each of those amino acids. In the leftmost helix wheel projection for troponin C, you can see that the vast majority of the amino acids within this alpha helix are charged in nature. The way that they're distributed around the helix is really unremarkable. There are a couple of spaces where there are hydrophobic, number 4 and 6 amino acids, but there's nothing special about that particular alpha helix. However, looking at the helix wheel projection for the protein on the right, alcohol dehydrogenase, we can see if we look at the primary sequence on the bottom and the chemical character of each of those amino acids, we have a hydrophobic, then a polar, then a charged, couple hydrophobic, a charged, couple hydrophobic. It doesn't seem particularly interesting. However, when we place each of those amino acids on the helix wheel projection, and they are now ordered as they would be in an alpha helix, we can see that the first, fourth, fifth, seventh, eighth, 11th amino acids within the alcohol dehydrogenase protein are aligned on a single side of the alpha helix. So there is a hydrophobic side of that alpha helix. The other side of the alpha helix is at the very least polar, and a few of the amino acids on that left side of the alpha helix are actually charged. This does look somewhat remarkable because it tells us that there's one side of the alpha helix that has one chemical character and the other side of the alpha helix that has a completely different chemical character. This can tell us something about the protein's function. This amphipathic helix might be found, for example, in a membrane, where the hydrophobic side of the helix would align itself with the hydrophobic fatty acid tails of the membrane, and the polar/charged hydrophilic side of the alpha helix might align with similarly hydrophilic regions of other alpha helices to create a pore in a biologic membrane. Alternatively, if we have multiple alpha helices that have hydrophobic sides, those hydrophobic sides are going to want to shy away, if you will, from the hydrophilic cytoplasm that would be inside of the cell. So it might find another alpha helix with a hydrophobic side, and those two hydrophobic areas would associate with one another. This indeed does happen. There are a subset of DNA binding proteins that are termed leucine zippers. Leucine is an amino acid that is hydrophobic, and if we find all of those leucines on one side of the helix, that helix can associate with another helix of the same chemical character and sort of zip up, as is shown in the middle image there. Another example of a coiled coil where hydrophobic regions of alpha helices associate with one another is the extracellular matrix protein collagen. The hydrophobic amino acid that is very prevalent in collagen is glycine. And if you remember, glycine is one of the smallest, or it is the smallest amino acid side chain. And that actually allows for 3 alpha helices to associate with one another, each of them with their glycine side chains hiding together in the middle of this triple helix. Of course, if all of the amino acid side chains that are sticking out from the surface of an alpha helix are hydrophobic, that might tell us that the polypeptide, or that region of the polypeptide with those hydrophobic side chains projecting, that wouldn't want to be in a hydrophilic environment. So it very likely would be embedded within a biologic membrane, as is shown here. So the alpha helix can be used to pass through a membrane, a helical transmembrane region, if all of the amino acids within that alpha helix are hydrophobic in nature. A single hydrophilic amino acid would not allow for this transmembrane function. The second type of secondary structure that can be found in polypeptide chains are beta sheets. And beta sheets are actually comprised of multiple beta strands. In the image that's on the right-hand side of this slide, we can see 3 different beta strands, and together those 3 strands would make up a beta sheet. Beta sheets can exist when there are 3 or more strands that associate with one another to create a sheet. Moving to the center image, you can see that the strands each sort of bend and fold in a zigzag manner in coordination with one another. Because of this, beta sheets are sometimes referred to as beta pleated sheets because they look somewhat pleated. There is one full pleat that occurs every approximately 0.7 nanometers within a beta sheet. And the amino acid side chains actually stick out from those amino acids. So I'm numbering the amino acids within one of the beta strands in this sheet, moving up from the bottom. As I said, the amino acid side chains are going to stick out from these pleats in an alternating fashion, such that the amino acid side chain of the one I've identified as number 1 will stick to the right, whereas the side chain for the second amino acid, or number 2, will stick out to the left. Number 3 to the right, number 4 to the left, number 5 to the right, etc., in an alternating manner. The amino acid side chains stick out or project from the beta sheet in this alternating fashion, again because the amino acid side chains themselves are not involved in the formation of a beta sheet. Instead, just like with alpha helices, hydrogen bonds between atoms within the polypeptide backbone are responsible for maintaining the structure, the secondary structure of a beta sheet. This liberates or frees the side chains to sort of not involve themselves with the beta sheet, and they are free then to involve themselves with other sorts of interactions. Just like with alpha helices, any amino acid can be found within a beta sheet with that single exception of proline. And again, that is because proline introduces a kink or bend within the backbone. And one might see how that kink or bend would interfere with the pleated nature of a beta sheet. Beta sheets themselves actually do come in two varieties. The first is shown here, anti-parallel beta sheets. Each of the strands of the beta sheets are oriented in alternating opposite directions, such that the amino end or the amino end of one strand will align with the carboxyl end of the next strand, which will align with the amino end of the next strand, etc. The image on the right sort of shows you how the amino acid side chains, those lollipop-looking things that are labeled with R, how they alternate sticking up and sticking down from the plane of this two-stranded beta sheet. If we look in the center, we can see that the amino end of the leftmost strand aligns itself with the carboxyl end of the next strand, which aligns itself with the amino end of the next strand, which aligns itself with the carboxyl end of the next strand. Hopefully that all makes sense. I've highlighted here the hydrogen bonds that are holding each of the strands together. The other variety of a beta sheet is parallel. And as you might imagine, in parallel beta sheets, the strands have their amino and carboxyl ends aligned in the same direction. This necessitates a non-sheet part of the polypeptide to sort of loop around, as indicated by those yellow lines there. So coming out of the carboxyl end of one strand, there's going to be a non-sheet loop that will find its way to the amino end of the next strand in the beta sheet. Hydrogen bonds also hold each of these strands together, as indicated on the left, but the hydrogen bonds look sort of wonky relative to one another. It may not be obvious, but it is possible for a polypeptide to have a beta sheet that is a combination of parallel and anti-parallel strands. So in this image on the left of a polypeptide that has alpha helices and the red beta sheet, you can see that 4 of the 5 strands within this beta sheet are in the same orientation, with their amino ends at the top and their carboxyl ends at the bottom. The exception is strand 4, which has its carboxyl end at the top and its amino end at the bottom. So strand 4 is anti-parallel to the other 4 strands that are parallel to one another. We can have really large beta sheets that actually wrap around to create what is called a beta barrel. Beta barrels are often associated with hydrophobic amino acids on one side of the plane of a beta sheet, a large beta sheet, and it's the hydrophobic amino acids that are being projected from that one face, if you will, of the beta sheet that will associate with the hydrophobic fatty acid tails in phospholipids of a membrane and create quite a large pore in a biologic membrane. If the chemical character of the amino acid side chains on both sides or planes of the beta sheet happen to be the same, it's possible for those beta strands to associate with one another in this sort of fibrillar fashion. So they stack upon one another with each of the faces associating with the faces of another beta strand to create this fibrillar form. The formation of these fibrillar structures by proteins with a large number of beta strands has physiologic ramifications. Here we see a soluble beta sheet monomer associating with other beta sheet monomers to create what is in the middle of the screen a beta sheet oligomer. In other words, multiple copies of a single type of protein, each with beta sheets, can have those beta sheets associate with one another so that the proteins stack upon one another to create what is in the next image, amyloid fibrils. These amyloid fibrils tend to accumulate both outside of the cell and inside of the cell. If they accumulate within the cell, it creates what's called inclusion bodies. In either case, a lot of these proteins associating with one another to create these fibrils that are oligomeric or many protein fibrils that accumulate inside or outside of the cell creates problems. Those problems are most frequently seen when this oligomerization by beta sheet stacking amongst multiple proteins is visualized in neurologic diseases like Huntington disease, for example, or in Alzheimer's disease, where the amyloid fibrils prevent normal neurologic function. Finally, while it might be obvious, I just want to highlight that it's possible for a protein to have multiple alpha helices, as shown in panel A, for a protein to have multiple beta strands to create beta barrels or fibrillar forms, as shown in panel C, and it's also possible for proteins to have a combination of beta strands creating beta sheets and alpha helices, as is shown in panel B.